All right. Okay, so I wanted to give you a practice question here. Again, I had a couple of people ask about this, so I wanted to make sure I was clear on the term. So in general, which of the following treatments is likely to make an antimicrobial chemical less effective? Okay, I'll give you a second to read that over. Think about what you choose. Okay, so let's see if we can narrow this down. So, of course, you always have to read the question. Am I asking for most effective, less effective? That's like a question that's like, which is the following are true, or which is the following are false? Of course, you gotta make sure you're looking for the right thing. So, less effective. So, A, if I dilute the concentration of the microbial, this, I'm not sure if I wrote this right. Oh, no. Okay, yeah, that actually is the correct answer. All right, but I didn't want to go through the right one first. All right, if I increase the temperature, yes, that's going to make an antimicrobial chemical usually more effective if we increase the temperature. So that's not right. If you use on a population of cells that's actively growing, usually when cells are actively growing, they're more susceptible. So that's not right. That's going to make it more effective. We'll see that especially when we use antibiotics. A lot of antibiotics don't work very well unless the cells are actively growing. Increasing the exposure time, that makes it work better too. And if we clean the surface before we expose it to the chemical, that is also going to make it more effective because remember, a lot of chemicals don't penetrate through organics. We talked about that organic load. Something like chlorine doesn't penetrate through organic load. That just means anything, some kind of cells or blood or even just dirt. A lot of chemicals don't penetrate through that very well. So we got to clean it up and then we can do our disinfecting. All right, so I didn't do those in the right order. I did that very anticlimactically, but yeah, if we dilute it in general, that usually makes our chemicals less effective. That's not always the case, but generally speaking. Okay, and then we had a couple questions last. Okay, go ahead, question. Antimicrobial, yes, thank you. Yep. Thank you very much. Okay. All right, and then we had a couple questions last time. They were good questions. I got home and I totally couldn't remember what the questions were, and I had to go back and watch my own video to remember what questions were. So one question we had was, why is hydrogen peroxide, why is it photosensitive? Why does it break down in the light? Well, if you look at the structure of hydrogen peroxide, it's got this H2O2. It's got this bond between the oxygen. It's called a peroxide bond. That's very weak. It's a very unstable bond. So this will dissociate by itself over time, but just being exposed to photons of light are going to make it even more unstable and dissociate even more quickly into water and oxygen. There are enzymes that will speed up that process as well. Some that you're going to, one in particular that you'll be testing for in lab in a couple weeks. So good question there. Hopefully that partially answers it. Another question about, can you use some kind of alcohol like vodka to clean a surface or maybe to clean out a wound? That may have some effect. And if I was Googling around and there are actually people that say you should clean with vodka, or say that you could clean with vodka. That's a lot more expensive to use that than a lot of other things. Typically, vodka is about 40% alcohol. So we talked about when you use alcohols, usually that sweet spot is between like 70 and 95%. So it's not going to work maybe as well as, other kinds of, alcohols that are made for cleaning. But you might have some effect there. It might be especially be effective against like a gram-negative bacterial cell or a membrane or a envelope virus, because those alcohols work pretty good on membranes. But again, because it's not as concentrated, maybe it's not going to work quite as well. And actually, wine is only 10 to 12% alcohol, beer like 2 to 6%. So they're going to have even less alcohol concentration in them. Okay, so I hope that helped to answer that question a little bit. Okay, all right. So we had our little kind of public health lesson last time, thinking about why we are surviving longer, why we are doing better in general against infectious diseases the last couple of years aside. I want to talk a little bit more history to start out with here, as long as some terms to make sure that we have straight. We'll talk about, in general, things that we have to think about when antibiotics are prescribed, like dosing, toxicity, allergies, things like that. And we'll get into, we have about 15 drugs that I want to talk specifically about, how they work and some uses. And hopefully you find this a little bit more applicable. If anybody wants to be going to pharmacy, hopefully you find it interesting. And we'll end with talking about antibiotic resistance a little bit, which I would say probably that's the part that students get a little bit confused about sometimes. Everything else, I'm just kind of dumping information on you. So I'll try to help you think about what's the most important things for you to kind of start going over and over. All right, so a little terminology. When we talk about something that's chemotherapy or a chemotherapeutic drug, usually we think about, oh, that's a drug to treat cancer. But in the broad sense, really, any chemotherapeutic agent is any drug that we use to treat a disease. And by disease, we mean injury to the host, to us, like something that's not that health state, I guess. So it doesn't necessarily have to be not an infectious disease, any kind of disease. And then if we're talking about treating an infectious disease, then we would call that an antimicrobial agent. So it could be an infection due to a fungi or a protozoa or a virus, anything. And then if we use the term antibiotic, okay, now we're talking about antimicrobial agents that target bacteria only. And traditionally, these agents have been biosynthesized, meaning produced by living organisms. So we'll get to what kinds of living organisms make antibiotics in a minute. Today, we actually do kind of monkey with some of those natural products, natural antibiotic products, and we can call those semi-synthetic antibiotics. So we still consider them, like I said, we still consider them antibiotics, but we have taken part of that molecule and changed it a little bit in the lab in order to give that drug some better properties, maybe make it be absorbed better or less likely for organisms to develop resistance to it. And then if a synthetic, we still consider them antibiotics if they, again, just target bacteria. So we have a couple examples of synthetic drugs that I'm going to talk about. Again, this differs. I know in your lab manual, they say, oh, it's not a true antibiotic if it hasn't been biosynthesized. It depends kind of where you get your definitions from, but I would consider any drug that only targeted bacteria an antibiotic, whether it's the natural product or whether it's synthetic. Okay. All right. So a little history lesson here. Paul Ehrlich, we talked about him before. He's kind of thought about as being the father of chemotherapy. So back in early 1900s, like 1905, he was testing a number, went through and testing a number of compounds, trying to find a drug that would be able to kill the organism that causes syphilis. And he found a compound that had arsenic in it that he called salvarsan. Paul Ehrlich, I believe, coined that term magic bullet. So he was searching for a compound that would kill the agent that causes syphilis and not be harmful to our bodies. Now, anything with arsenic in it is going to be harmful to us. So it had very limited use. It was maybe, partially effective there in really small doses, but it was a start. It was a start. And really, we didn't find any other agents that were useful against bacteria until the 1930s, where Gerard Domack, I think I'm getting the pronunciation close there. He was a German scientist that was, again, systematically testing a number of compounds against streptococcus in mice. And he finally found one that he called, I'm blanking on the name of it, but it was a sulfa drug, Prontosil was the name of it. And sulfa, it was a compound that was really quite cheap at the time. It was used for a lot of other things, so they kind of struggled to figure out how to market that so that they could make some money. But again, this was useful against streptococcus. And that was a really big deal. It was really only useful against that, but there was a lot, strep throat, scarlet fever caused by streptococcus, which caused really high mortality at that time. So those drugs were credited with saving the life of Winston Churchill before World War II. So that was a big leap. He was a German scientist. He won the Nobel Prize in 1939 for this, but Hitler wouldn't let him go to accept his Nobel because he was a pacifist. And so he didn't get his money, but he did win the Nobel for that. And then in 1929, of course, Alexander Fleming, one of my lab sections got a big laugh yesterday when I said Alexander Hamilton by accident. Alexander Fleming, that's him right here looking very scholarly. The story goes that he left his lab and went on vacation for a couple weeks. And back then, when you grew up your organisms in lab, you didn't have a bunch of plastic everything. They had glass petri dishes. So he left his dirty dishes sitting there, his old bacterial cultures in his glass petri dishes. He went on vacation for a few weeks. And when he came back, he noticed one of his plates of staph, it was some kind of staphylococcus he was working with, looked like this. There was a big contaminant growing on that plate. And he noticed, hey, this contaminant, this mold is secreting something that's inhibiting the growth of that staphylococcus. So he was already experimenting with lysozyme, which is an enzyme that breaks that peptidoglycan wall of bacteria. He was already doing experiments with that, trying to figure out the ways to maybe use that as a drug. So his mind was kind of primed for this. And he set out to try to isolate the substance that he called penicillin, because this was a penicillium mold. And he was able to isolate it, but he wasn't able to get a big enough quantity of it. He couldn't get the mold to produce a big enough quantity of it to be useful to treat anyone. So he kind of wrote it up in a paper, and then he moved on to something else. Well, in the 1930s, and he did his work in London at St. Mary's Hospital, I think, Alexander Fleming. And then in the 1930s, Howard Florey, that's him right here. he started systematically looking at the literature, trying to figure out something that would be useful against bacterial infections, because he saw war brewing and he said, we need this to save lives. So he assembled a team around him, including Ernst Chain. That's him right there. Chain was a organic chemist, and he found Fleming's paper and said, let's see if we can make this work. So they set about trying to figure out how to do this organic synthesis to extract enough penicillin. They had to grow up the mold in great big vats. And finally, we're able to figure out a way to mass produce penicillin. And it was really available within a few years there in the early 1940s. So what else was I going to say about that? Okay. Oh, so apparently you can only have 3 people sharing a Nobel Prize. So, you know, a number of people worked on this mass producing penicillin, but it was Florey, Chain, and then Fleming that shared the Nobel Prize, even though Fleming really, he had like just the idea of it, but didn't really actually make the actual drug. Okay, what kind of, yeah, question? It was some kind of staphylococcus. And in fact, penicillin only works on gram-positives, or at that time, the very first penicillins. Yeah. All right. So I alluded to this already. Where do antibiotics come from? There's several genera of bacteria that secrete antibiotics, as well as various genera of fungi. Okay, so no, like, you know, plants don't secrete antibiotics. We don't really know why these organisms secrete antibiotics. It seems like maybe they would help them outcompete other organisms in nature if they're killing other organisms around it. But we think in nature they don't actually secrete high enough concentration in order to do that. So we think it could be some kind of cell signaling function. Bacillus, that's a genus, again, that secreted a lot of antibiotics. This Streptomyces is a genera of bacteria that kind of acts like fungi. It grows, that's what's pictured here. It's grown in these long, thin filaments, and it actually produces spores that are reproductive, like fungi. Yet they are prokaryotic cells. They don't have a true nucleus, don't have organelles, so it's kind of one of those weird organisms. Any organism that makes an antibiotic, I believe all of them are found in the soil. Okay, so Bacillus subtilis, that's an organism that you probably have on the bottom of your shoe right now if you've walked outside today. Okay, soil organisms, of course, the most famous fungi, the penicillium mold, that's what's pictured here. It usually has kind of a greenish tinge to it. And then this is an acremonium mold that used to be known as cephalosporium. So we talk about that in our lab manual, and we call it the cephalosporium when actually they changed the name of that genus. So now it's acremonium, which is really confusing because we have all these antibiotics called cephalosporin antibiotics, and it makes sense that they come from a cephalosporium mold, but I'm not sure why that name was changed. But generally when molds are growing up on a plate, they look kind of fuzzy, okay, and kind of grow great big colonies like that. All right, so let's talk about, again, before we get into kind of our run of drugs, when we're thinking about what kind of antibiotic to prescribe, or when we're trying to develop new antibiotics, really the first thing that we have to consider is that drug selectively toxic? Okay, so in other words, does the drug target pathways or structures that the host lacks? Okay, this is the magic bullet concept. Do we have a drug that's going to harm the invading pathogen, the bacteria, and not hurt the host. Okay, if a drug is not selectively toxic, then it has very limited use because, again, we don't want to do any more harm than what's there already. Okay, and that's really the hard thing today. We've got lots of antibiotics that we could be, new ones that we could be turning out, but the problem with a lot of them is that they're too toxic for us to take. So that's kind of the primary thing that I want you to take away from today. When we start talking about mechanisms, think about how they're selectively toxic. Okay, so lots of other things that we need to consider. The spectrum of activity. If it's a narrow spectrum drug, that means it targets only certain bacteria. And this is going to be good to use if you know what's causing the infection, if you know what agent is causing the infection. Other drugs are going to be more broad spectrum, and that just means targets a wider variety of bacteria. For instance, maybe gram positives and gram negatives rather than just, gram negatives. Oftentimes we might use a broad spectrum antibiotic if we're trying to use a drug preventatively. Another way to say that is to use it prophylactically. So prophylactic use. So for example, my son went and got his wisdom teeth out. At the same time, the oral surgeon gave him a couple doses of penicillin to take. Our mouth is a pretty dirty place. We have a lot of bacteria in there. So just to kind of ward off the chance that there's going to be an infection from getting those wisdom teeth out, he took a couple penicillin actually. Also, we might use a broad spectrum antibiotic if a patient has a mixed infection, meaning they're infected with several different kinds of organisms at once, numerous species. Now, using a broad spectrum antibiotic does have its risks. Okay, we're going to talk much more about normal microbiota in the next coming chapters, but it is, when I say normal microbiota, I mean the normal bacteria that are just residents within our body that aren't doing us any harm. So it's going to impact our normal microbiota. Again, especially when you take a broad spectrum antibiotic that's going to kill a lot of different kinds of bacteria. And that when your normal flora, maybe some bacteria maybe are knocked back and, not going to be there as present in numbers that they usually are, that's going to potentially cause what we call a super infection. And that just means one organism that's normally part of your normal microbiota in small numbers is allowed to proliferate more than what it normally does because other organisms in your normal microbiota are killed. Okay, so let me write that down. So that just means some species, I didn't leave myself really enough room here, of normal microbiota allowed to proliferate. Okay, and that can cause disease. Sometimes we call a superinfection a secondary infection. Okay, so some examples of this, here, this lovely picture that I have here, thrush. Okay, that a lot of us have just normally maybe a small number of yeast in our mouth. And if we go on maybe a course or two of antibiotics that knocks out some of the other bacteria in our mouth, the candida yeast can overgrow and give this kind of, whitish growth, which is kind of nasty, not super dangerous or anything. Another example might be a yeast infection, a vaginal yeast infection. Okay, again, common if somebody's on broad spectrum antibiotics. A third example, Clostridium difficile, C. diff. Sorry. Whoops, didn't mean to do that. Okay, we have a lot of us again have Clostridium difficile in our colon in small numbers. And again, if that variety is changed a little bit, maybe the C. diff can overgrow, and that causes a really nasty diarrheal disease, enterocolitis, that can be almost really debilitating, almost an ulcerative even. So just some examples, again, of super infections, why sometimes we may want to avoid giving a broad spectrum antibiotic if at all possible. Sometimes it can't be avoided. Oh, okay, so I meant to show this before. When I was talking about selective toxicity, here's some examples of targets that microbes, that the drugs will have so that they're selectively toxic. Okay, cell wall inhibitors. Don't worry about the drugs that are listed here. We're gonna talk about some of them, but not all of them. That's a really, we probably like half of our antibiotics target the cell wall of bacteria, and that's good selective toxicity because, of course, we humans as animals, having animal cells, don't have cell walls. So again, something is targeted that we don't have, good chance it's going to be less toxic to us. A lot of our antibiotics, a huge class, inhibit protein synthesis. And remember, bacteria have a slightly different ribosome structure, 70S, than eukaryotes. We have 80S. Not as many choices when we think about drugs that target the cytoplasmic membrane, because the membranes of bacteria are very similar to our membranes. So again, a lot of toxicity issues there. There are a few metabolic pathways that bacteria have that we don't, that we can easily target. And then DNA replication and RNA synthesis. Again, some of the enzymes there are different enough that we can get drugs that target those and not bother us. No, don't memorize these drugs, not yet. No. In this class, we're not going to talk about those are some antivirals, so we're not going to get to that. Yeah, so just kind of thinking about the mechanisms why they're selectively toxic or not very selectively toxic for now. Okay, a few more things to think about, especially when we're thinking about which antibiotics to prescribe. Consider the toxicity. We have a way to kind of tell the toxicity of a drug by comparing the dosage that a patient can tolerate to the dosage that will actually kill the microbe. Okay, we call that the therapeutic index. Okay. So the goal is we want to select the dosage that's going to be really effective without harming the host, obviously. And the difference between those two things we refer to as the therapeutic index. So, for example, if the maximum dose of drug that a patient can tolerate, say that's 100 micrograms, okay? And so we might call that the toxic dose. That doesn't necessarily mean the dose that's going to kill a patient. It's the level that the dose where the side effects become unacceptable. So for example, if you're trying to treat somebody that has cancer, if the drug makes their hair fall out, okay, that's just that in that case is an acceptable side effect. you have a drug to treat an ear infection and it makes your hair fall out, then that is not considered an acceptable side effect. So again, that's the point, that the side effects become unacceptable, and compare that to the dose that the drug works and kills the pathogen. So say 100 micrograms is the toxic dose, and it only takes 1 microgram to kill the pathogen. Okay, so then the therapeutic index is going to equal to 100. Okay, that's a fairly acceptable, that's considered a very safe drug. Okay, so the higher the therapeutic index, the safer the drug. When we start getting down, and I've tried to look up, therapeutic indexes for a lot of different drugs, and it's not something that's easily available. The problem is that what's safe for one person can be a little bit different than what's safe for someone else. So they kind of have to try to test this, hopefully on an animal, and have a wide range of doses to try to figure that out exactly. Okay. We also need to consider the half-life of a drug, meaning how quickly does the concentration that's actually going to work to kill the pathogen, how quickly does that dissipate? Okay, so if a drug stays or degrades really slowly, then you don't have to dose it as often. And that's kind of convenient. Also, the administration method of the drug. So ideally, a drug can be given orally, and that just means it's swallowed. Okay, certainly that's the simplest way for people to take drugs at home. Some drugs can't be given orally, and so we have to give them parenterally. That's a word that you're going to see later on as well. That means not swallowed. That root enter or enteric, enteral, that means of the intestine. And then par means against or not of the intestine. So if we're not giving it sort of the normal way of swallowing it, then we have to either give it intravenously, so by IV, or injected. Sometimes we call that intramuscularly, IM. Okay, and some drugs, some antibiotics are also given topically. Okay, usually for skin infections. Okay, so why can't some drugs? Like I said, some drugs we can't give orally. So some drugs are going to be affected by stomach acid. And we call those acid labile. Okay, that means they lose their activity or their structure changes in the presence of our stomach acid. Stomach's a pretty acidic place. It's like pH of 2. Some drugs are just not absorbed well in the small intestine, through the small intestine. So some drugs not absorbed well. Through the small intestine. So that's another reason that you might not be able to, or might not want to give them orally. And in addition, if a drug has a very low therapeutic index, then we probably don't want to trust a patient to take it orally, because they might not take the drug at the right time. It might not, if they take the doses too close together, you might get too concentrated. So in addition, if the therapeutic index is low, it's always going to be given by IV. Okay, because when we give a drug intravenously, we can dial in that concentration to whatever we want. So if it's going to be really close to a toxic dose that needs to be given, we're going to give it by IV. Question? Yes. So, like if it's a very toxic drug. Sorry. Yeah, I think so. I think because, right, and that's going to be done under hospital supervision if it's injected, and so they can dial that in a little bit more carefully. Okay, so this chart just kind of, when we inject it, if we give the drug by IV, the concentration comes up really, really quickly in the blood. And actually, we can drip that in. So it's staying at high concentration. Sometimes that's necessary. Usually we inject it. That's the green line here. That's going to come up to concentration more quickly than if we swallow it. And again, if it's kind of toxic, we don't want to let the patient, we don't want to let the patient maybe make a mistake in terms of when they take it or not. So all things to consider when thinking about how to administer a drug. Also need to think about interactions. Like obviously sometimes when you give two drugs, they're gonna maybe be harmful interaction. We would call them antagonistic. Sometimes we might always give two different drugs together because they work together and bring the patient back to health much more quickly. And we'll give some examples of that in a little bit. One example is our sulfa drugs. We usually give sulfa drugs in conjunction with trimethoprim because they target different enzymes in the same biosynthetic pathway for folic acid. We might have talked about that in lab this week, maybe not, but we'll talk about that again later. Drugs can be antagonistic for all sorts of reasons. You don't want to give an antibiotic that keeps bacteria from growing along with an antibiotic, that penicillin, that only works on actively growing cells, right? They're going to kind of cancel each other out a little bit. Some antibiotics are known to depress the function of birth control pills. So, there's just all sorts of things that have to be considered there. Also, are organisms susceptible? If you try to treat an infection of a bacteria that doesn't have a cell wall and you give them penicillin, it's not going to do any good. That organism is just going to be naturally resistant to that. And we can test whether an organism is susceptible or resistant to an antibiotic by doing a Kirby-Bauer test. So if you're in lab, you did this yesterday or the day before, but for those of you not in lab, this is a Kirby-Bauer test. Basically, if you have a patient maybe that's struggling with some infection, you're trying to figure out what antibiotic would work best, you perhaps could grow that pathogen up in pure culture. If you were able to do that, able to figure out what the organism was, you would make a lawn of bacteria on a certain kind of media so that it's a very standardized process. And then try various, these discs have the certain antibiotics impregnated in them. You incubate, and then you look to see how big the zone of inhibition is where the organism could not grow because that antibiotic was diffusing out in the medium. In general, you got a big zone here, then that antibiotic is probably working pretty well. There is a little difference. We take into account the diffusion rate and things like that. We have standardized charts to compare how big the zone needs to be until it's considered susceptible. But if the organism is growing right up to the antibiotic, like right here, then that's antibiotics not doing any good. Okay, so that's a test that's been around for a while. Okay, so then we start thinking about the mechanisms. And when I went through this today, I realized, oh, this is pretty much the same slide that I showed you a little bit ago that listed all the drugs and the different mechanisms. So this shows the targets of the bacteria cell, but it's pretty much the same thing. So again, that's just your OpenStax picture. Okay, so I know we have a few minutes left. Let's start thinking about a huge class of drugs that are in use quite a bit still. They inhibit cell wall synthesis. And it's a class of drugs known as the beta-lactams. Okay. Of course, penicillin is a beta-lactam. That's one example. Methicillin, that's another drug that we're going to talk about. There's a huge class of beta-lactams called carbapenems. We're not going to worry so much about those. We're going to mention the cephalosporins a little bit later. But all these drugs have in common here, they have this beta-lactam ring. So that, if you're interested in the chemistry at all, why do we call them beta-lactams? This ring it's a cyclic amide, if you're interested, and it's beta-lactam because it's the beta spot that has that nitrogen right there. Beta means like the second one from the carbonyl group right here. So that's why they're known as beta-lactams. What they all have in common is that beta-lactam structure. That's why they've colored it in green there. If that beta-lactam ring is cut or changed in any way, then the drugs lose their ability to function. So we'll talk about why here in a minute. I like this. This is a graphic from our old textbook, and it shows a little bit how they inhibit peptidoglycan formation. All the beta-lactam drugs bind irreversibly to the enzymes that cross-link the NAM subunits. So remember the structure of peptidoglycan, it's NAG, N-acetylglucosamine, and N-acetylmuramic acid, NAM, alternating sugars with some amino acid bridges in between. Those amino acid bridges are in the growing cell wall, are made by enzymes called transpeptidases. And we talked about that when we were talking about cell structure before, so maybe that rings a bell. Other enzymes called autolysins are going to cleave those bonds between the sugars, but it's the transpeptidases that they're going to make these new cross bridges. Again, when the cell is adding new NAM and NAG subunits in order to grow its cell wall. Okay, so normally when the cell grows, the new subunits here, they're outlined in white, they're added in, and then you get these new amino acid bridges that are formed by the transpeptidases. Okay, if, however, the cell is in the, has beta-lactam antibiotics present, that's what those little green dots are supposed to be, then this is showing where the cross bridges should be, and they don't get formed. So the green is where they should get formed after the new subunits are put in. And because the drug are bound to this enzyme that forms those bridges, they don't get formed. And so now the cell wall is all kind of wonky like this, and it's going to be susceptible to osmotic forces. When the cell's in a hypotonic medium, water rushes in and that cell wall is not intact enough to keep the cell from bursting. So it's not really the drug that bursts the cell, but it's the osmotic forces. Okay, so we'll keep on going with our antibiotics next week. If you haven't been working on your virus homework and the microbial control, be nice, cold, rainy this weekend, good time to do some homework. Have a good weekend.